A patient with beta thalassemia major undergoes a clinical trial for gene therapy. 2 years later, he develops acute myeloid leukemia with evidence of clonal expansion of a transduced hematopoietic progenitor population. Which of the following characteristics of the gene transfer vector most likely contributed to this patient's condition? Choice A, induction of apoptosis from viral regulatory genes. Choice B, transient expression due to non-integration. Choice C, insertional activity near enhancer sequences. Choice D, host immune response to viral vectors. Or choice E, lack of replication potential amongst vectors. I'll wait 5 seconds and then answer the question. Sometimes USMLE likes wrapping up basic biochemistry questions in these weird questions about experiments and whatnot. But when you strip it all down, it should be a pretty easy question for most people, because what it's really asking is, what can cause a malignant change in a cell? All this stuff about viral vectors and whatnot is just window dressing. And of course, USMLE wants you to understand how proto-oncogenes work and how DNA, RNA replication works, and arguably to some degree, I guess, an understanding of how viral transduction works. But the answer is pretty simple, right? Choice C, insertional activity near enhancer sequences, is the correct answer, because you just need to have the general understanding that promoter regions or enhancer sequences are usually near the beginning or right before the actual translated sequence in DNA. So if there's some kind of weird change that upregulates gene transcription, that can lead to malignant change. Nothing complicated. And even without that, usually all the other answer choices on these kinds of questions are so obviously wrong if you really just sit down and think for 5 seconds about what they're saying. Choices A, B, and E, induction of apoptosis, transient expression, and lack of replication potential, just thinking about it from an intuitive standpoint, these would cause the opposite effect, right? They would more than likely lower the chances of malignant change in a cell. And then we have choice D, host immune response to viral vectors, which is just to say that sometimes viral vectors can cause inflammation. And maybe you can sit around and logic out the immunology of it, but really all that means is that you'll have inflammation and maybe destruction of virally infected cells and that sort of thing. And sure, you can maybe argue that, well, chronic inflammation could arguably lead to malignancy, right? But that's just obviously a much worse answer choice than just saying that there's some kind of change to the genetic sequence as a direct result of you introducing a viral vector of some kind. Easy question, but still got to talk about it because this sort of question actually does crop up fairly often. I hope you found this video helpful. I'll see you in the next one.